Okay, good morning, everyone. First off, I'd like to give my condolences to the Laker fans out there. Just saying Kobe Bryant met his match with some more extracellular matrix. Just happened to snap on him during the game. I'm sort of an anti-Laker fan, so that's how like it is from my point of view. Anyway, hope you guys had a good weekend and ready for yet another wonderful week on Cal. Today, office hours are noon to 1 in 28.4 BLSB. My other office hours are Thursday 2 to 3. Also, you can always email me questions. If they're rather specific questions, that's totally fine. If it's repeat lecture 2, I'd say go to the webcast, that kind of thing. But if you have specific questions, email works. Today we'll finish up on our start insulin receptor action, glucagon receptor action, and there's questions on pages 30 to 31. You should, after today's lecture, you should try working on them. And then we'll hit starting on male reproduction, spermatogenesis, and negative feedback. I don't know how far into that we'll get today, but it'll be the next topic. Okay, so in terms of review, we talked about hormone agonists versus antagonists. And I wanted to point out, there's a few questions from the lecture on Friday. I just want to point out that you can have two different hormone agonists that have opposing effects. They're not antagonists. They are agonists that have an opposing effect on a process. An example is glucose homeostasis, or insulin brings down glucose levels, glucagon brings it up. They are not antagonists, hormone antagonists. They are hormone agonists that have an opposing effect. Whereas a hormone antagonist, the way we're referring to it, refers to the mechanism of action. There are several classes of antagonists, but the one class I want you to remember is that you have receptor binding and prevents the agonist response. A lot of examples, one I gave you was RU486. The attachment of the placenta is a progesterone hormone response. The abortion pill RU486 is a progesterone receptor antagonist. So it binds the receptor, prevents progesterone from binding, and causes detachment. And by the way, there's a question about the morning after pill. Turns out, I looked that up. I didn't actually know precisely what that was mechanistically. Turns out the morning after pill is actually two hormone agonists, estrogen and progesterone, that together prevent ovulation and prevent fertilization, is the primary means by which it works. So there you have two hormone agonists coming together to do something. Okay? And a lot of times it's more than two hormones that affect response. Does everybody see the difference between opposing actions of two agonists versus mechanistically what we're referring to as an antagonist? Okay? I guess from a literature standpoint, you could say, yes, two opposing hormones have an antagonizing effect on each other. Well, let's not mix it up. We'll just call it opposing actions. Might be the best way to think about it from a mechanistic standpoint. Okay? And a lot of medicine is based on exactly this concept. Hormones acting as agonists versus antagonists. Lots of different stuff out there. Okay? Any questions on that? Very important to see that difference. Okay, so we talk about steroid hormone action. Steroids bind, go through the plasma membrane, being small, greasy molecules, bind to receptor. That receptors are transcription factors, sits down on the DNA in the promoter region of a gene. It's not the only place they sit down, but it's the only place you guys need to know about for now. Receptor binds a steroid. Receptors bind a transcription factor or another protein on the gene, and receptors bind DNA. And those three binding interactions dictate specificity. Steroid binding, transcription factor binding, DNA binding. And we talked about androgen insensitivity syndrome. These are genetic males that look like females in terms of external genitalia. They have defects in steroid binding. They have the androgen receptor, defects in transcription factor binding, or simply mutations. And mutations in DNA binding. In addition, there's mutations in expression of the receptor in the first place. Because these receptors are proteins, they're encoded by genes, they have promoters, and you can have lack of expression. And this kind of syndrome kind of cemented the issue of the importance of all three binding interactions of the receptor. And there are others out there. This is one of the more dramatic ones. Okay? Then we talked about gel shift assays. This DNA binding assay where these molecules electrophorease through the gel based on size. So the DNA alone is the only thing you can see. And it migrates to a certain level, to a certain place on the gel. When the receptor binds, the band is shifted up, so it's called a gel shift assay. So when a GR binds a GRE, glucocorticoid-regulated elements, it's a sequence-specific binding that we talked about. You do not need to know the sequence. You shift up the band. If you put in the estrogen receptor, which binds to its estrogen response element, the band doesn't shift, so therefore it's not binding. So you shift up the band binding, no change in the band from the pure DNA, no binding. If it shifts up, there is binding. Everybody see that? The unusual feature in the field was that the progesterone receptor, the androgen receptor, and the mineralocorticoid receptor all bind the glucocorticoid-regulated element. So these four receptors, GR, glucocorticoid receptor, AR, progesterone receptor, androgen receptor, and mineralocorticoid receptor, all bind the same DNA. So how do you get specificity? And to expand on that just briefly, so you can see this, the DNA binding is the same. So the specificity must be in the steroid and the transcription factor binding region. And in endogenous genes, you have different sets of transcription factors. So the glucocorticoid receptor, it has its transcription factors it interacts with on glucocorticoid-regulated genes. The progesterone receptor, for example, has its transcription factors on a different set of genes. So that dictates the specificity. And there are some thought in the field that the most important part of specificity is the transcription factor interactions. In addition to physiologically, you've got to have the hormone around. And the target cell must express receptor. So in a cell that expresses both glucocorticoid receptor and progesterone receptor, the difference in response will be the presence of specific transcription factors on the gene. Does everybody see that? And each of these genes has their own constellation of transcription factors, which not only change, just as an aside, not only change specificity, but change magnitude of response. And in some cases, whether response goes up or down. Okay? So these transcription factors do a lot. Questions on that? Very important point. AIS? Very good question. The question is, and there's a very complicated answer to that, and I'll give you the short version. The question is, because these transcription factor interactions are so important, would these mutations be the more severe phenotype? Maybe that's what we're trying to say. It turns out it's actually the largest range of phenotypes, from the least severe to the most severe, because there's many kinds of protein receptor interactions. So some of those might be in a place that's just a little bit important. So you get, say, a sterile male that comes out of it, whereas others that might be incredibly important, but the surfaces are big, so you have more chances for different kinds of mutations. That's a very good point. And people don't realize that right away, that you get a range, which would be predicted based on the kinds of interactions you might see there. Yeah, a lot of people study that. It's a very important point. Okay? Now, then we talked about, we shifted gears to cell surface receptors, which we'll continue today in about a minute or so. And you have an extracellular domain where the hormone binds, you transduce a signal through the receptor, and this has to do with changes in the way the receptor interacts with itself and with other protein components, which we'll get to. And then you get signaling. And what I'd like to discuss, there's a lot of different protein hormone receptors. But we're going to discuss just insulin receptor and glucagon receptor, because they represent two of the most well-studied molecular mechanisms, by far. And they work in opposing actions on glucose regulation. So those of you who ate breakfast this morning, I imagine the same group that had breakfast in the morning on Friday. Most people stay in the same kinds of habits, either they eat or they don't eat in the morning. Increasing glucose depolarizes the membrane of beta islet cells in the pancreas, causes an influx of calcium, which causes insulin to be secreted, binds to a receptor in the liver. The liver then transports in the glucose, stores it as glycogen or uses it. And that reduces glucose levels in the blood. Those of you who didn't eat yet this morning, assuming you didn't have dinner at 5 in the morning from staying up all night, so you're low on glucose. This signals the alpha islet cell to secrete glucagon, a different hormone, which then goes to a receptor, its set of receptors in the liver, and causes the synthesis and secretion of glucose. These two hormone agonists work by fundamentally different mechanisms, but they are prototypical of two extremely important classes of receptor mechanisms, and that's why we'll just discuss these two. Again, there's lots of others. We're just going to focus on those two. Okay? Questions on glucose metabolism before we get into this? Again, insulin and glucagon are both agonists, but they have opposing effects on the level of glucose. Okay? All right, so we're going to start with insulin. Let me put this down here. Insulin is, putting these boards down for a reason. Insulin receptor, it typifies the kind of receptor that's a single pass transmembrane receptor. And in the generic, this is the generic part of this. So this is the generic pathway. Now, the insulin receptor is a variation of the theme. So you need to know the theme before we can discuss the variation. It's part of a class of receptors that are single pass, that was one time through the membrane, transmembrane receptors. And it has an enzyme in its structure. So these receptors are basically hormone-activated enzymes. So here's the plasma membrane, and this is the generic. It's sitting in there, and it has an enzyme in the cytoplasmic tail. So this is the inside of the cell, or the cytoplasm. This is the outside. And this, put this up here. This enzyme is a tyrosine kinase in this class of receptors. If you see the word kinase, what does this mean? You guys should have had this earlier on. It phosphorylates stuff, right? What do you think it phosphorylates? Something with a tyrosine. Exactly. And so basically you have some other molecule like a protein in the presence of ATP, the tyrosine kinase, which these receptors are, then phosphorylated. Okay? Knocks off a high energy bond on ATP. This is a critical component of how this receptor works, is to understand this enzymatic reaction. Because if I said to you, what does every self-respecting enzyme must have? What should be your answer? A substrate, right? And that's the key to understanding how it works, the substrates. Okay, so first we're going to do the generic part of the pathway. I'll put the boards up, and then we'll do the insulin receptor variation. So the actual set of receptors are growth factor receptors. And the one that's usually used is EGF, or epidermal growth factor. It's actually a very important one in cancer biology. A lot of the signaling of this receptor is a target of several kinds of very important anti-cancer drugs, for example. So it is an important pathway. But the way this works is, first you have this receptor sitting in the membrane. This is the plasma membrane. And this is inactive. Let's put it down here. So this enzyme is inactive. The hormone binds and then causes receptors to dimerize. Okay, so you get a structure that looks like this. And this is, dimerization takes place on both the intracellular and extracellular. So my two erasers here, which will be receptors today. So this dimerization, what this does is it activates the tyrosine kinase. So the enzyme on this receptor is inactive until the receptors dimerize. There's a conformational change, a reaction. And the way, of course, I have a little bit of asthma, but I'll do it over here. So these receptors come together. The hormone binds. There's a reaction. See, you can get a reaction just by binding. Hopefully nobody has as bad asthma as I have, so I'll do it over here. So the point is that coming together changes the conformation in a way that now activates the tyrosine kinase. So the question I have you again is, what does every self-respecting enzyme must have? A substrate, right? Well, it took a while in the field to figure this out, but it turns out for this class of receptors, the substrates are the other receptor molecule. So you get receptor phosphorylation at tyrosine residues. These receptors have enough of tyrosine residues. And there's very specific places in which these are phosphorylated. Everybody see that? The substrate for the receptor is the other receptor subunit that comes together. There's a variation of that with insulin receptor, but not by much. So then what's the reason for this? These phosphorylated residues then become landing sites for cell signaling molecules. And these cell signaling molecules range from other other enzymes to other kinds of molecules that link a whole bunch of proteins together and create a cell protein complex right under the cell surface. I'll just do this for one of the receptor molecules. So these signaling molecules come in and bind. And there's ones that bind at very specific places. So if a receptor has, you don't have to know this, but if you have 3 different phosphorylation sites, you'll have 3 different signaling molecules bind very specifically at each site. And then the result of this binding kicks on a response. And these signaling pathways can lead to growth. They can lead to changes in all sorts of transcription factor expression, all this stuff. Okay? But the way to view this is of a specific sequence of events. Inactive receptor, hormone binds, receptor dimerizes, activate the enzyme that's in the receptor itself, phosphorylation of the other receptor subunit, which for a lot of these growth factor receptors is the only known substrate. Cell signaling molecules bind to the phosphorylated region of the receptor. Without the phosphorylated region, those cell signaling molecules will not bind. So that's how you kick on a signaling pathway. Then these signaling molecules on the inside of the cell kick on a whole array of pathways, which you don't need to know about. And if you're interested, I teach a hormone action class in the fall that spends about 4 or 5 weeks on this. Okay? The point is, it's a way in which a hormone signal outside the cell gets transduced inside the cell. Questions? Yes? Wait, say that again? I'm still not sure your question. The tyrosine kinase is in the receptor itself. The cell signaling molecules are other kinds of enzymes. One being, as an example, PI3 kinase, which is a serine-threonine kinase. There's other kinds of molecules. These cell signaling molecules are just hanging out inside the cell. And when they see a phosphorylated receptor, they see it as a high affinity landing site. And that interaction, that receptor-protein interaction, activates the cell signaling molecule. Right. As an example, kicks on other pathways. That's exactly right. So let me say to you, what if the tyrosine kinase was dead? What would you predict about this pathway? Yeah, if it was dead, it would be inactive, and therefore what would not happen if the tyrosine kinase was dead? you never get phosphorylation, right? And if you don't get phosphorylation, guess what? You don't get the cell signaling molecules binding. Let me rephrase it. What if the tyrosine kinase was constitutively active, always on, regardless whether there's a hormone? What would you always see? Receptor phosphorylation, and what would you also see after that? Binding the signaling molecule. That's precisely one of the problems in breast cancer. There's a growth factor member of this family called HER2, which is always active. And they developed a drug to actually inhibit that particular receptor. So it's out there. Okay, this is important stuff in terms of these pathways. Okay, a series of events. Everybody with that? If the tyrosine kinase is always on, again, guys, look at the questions over there. There's some questions on it. If the tyrosine kinase is always on, you always get phosphorylation, and you always get cell signaling molecules bound. So now let's look at this is the general class of these receptors. Insulin has two very important variations. So now this is more related to insulin receptor. Its variations are as follows. The insulin receptor always sits initially in the inactive state in a very loose dimer, or heterotetramer, or just view it as a dimer. This is the enzyme. It's a tyrosine kinase, and this is inactive. It just happens to be in a loose dimer, held together by disulfide bonds. So we'll worry about that. Upon hormone binding, instead of dimerization, it's already in sort of a dimer. What you have is something called clustering. So you get receptor clustering. And that just means you get a bunch of them that come together. And it's this clustering, I'll put this down in a second. This clustering is what activates the tyrosine kinase. Hey, everybody see that? So instead of a dimer, you get a cluster. Mechanistically, it's the same thing. You do not need to know the difference between dimers and clustering. Both cases, dimerization for growth factor receptors or clustering for insulin receptors activates the tyrosine kinase. That's one variation. That's important to think about. Any questions on that? The other variation is, let me ask you again this question. What does every self-respecting enzyme must have? Substrate, right? Well, for insulin receptor, there is an interesting variation of this theme. Here's the insulin receptor, the active kinase. And in fact, one of its substrates is the receptor itself. This is IR for insulin receptor. So one of them is exactly like the growth factor receptors. But in addition, there's another substrate that they found, actually family receptors. I'll put the board down here in a second, called insulin receptor substrates. And one of the first ones found is the important one. And given the fact that it's April 15th, it's kind of appropriate to say the acronym for this particular molecule is IRS, insulin receptor substrate. Okay? And insulin receptor substrate comes up in the presence of ATP, and where do you think it's phosphorylated? If this is a tyrosine kinase, on tyrosine residues, right? And IRS, the insulin receptor substrate, have many tyrosine residues. So for insulin receptor, you not only have phosphorylation of the receptor itself, but you also have phosphorylation of IRS1, the insulin receptor substrates. And in both cases, here's the receptor. Here's one of the phosphorylations. Cell signaling molecules bind, and cell signaling molecules can bind to IRS to then together kick on the response. In some cases, the same signaling molecule that binds the receptor is the same one that binds IRS1, if this landing site is correct. What you view these IRSs, insulin receptor substrates, are as an expanded platform of the receptor. The receptor has about 4 or 5 approximately important phosphorylation sites. IRS1 has another 10 to 15. So you create a larger landing site for these substrates, for the signaling molecules to bind. So you're expanding the platform of the receptor, is the role of these. IRS1 is the most important one, but there are others. Questions on that? Does anybody see the differences between the growth factor pathway and insulin receptor pathway? Almost very similar, except you've got two interesting variations. Yes? What is the difference between a substrate and receptor? The receptor is an enzyme. In its structure is a tyrosine kinase. In its structure. What does every self-respecting enzyme must have? A substrate. Well, it's a substrate. It turns out the receptor itself is also a substrate. The other subunit is a substrate. So the receptor, insulin receptor, has two very important classes of substrates. The other receptor subunit and these IRS family of molecules, insulin receptor substrate, i.e. the name. They didn't name it after the Internal Revenue Service. Okay? Very important to see that. Receptor itself is an enzyme. Just like steroid receptors are transcription factors, these guys are enzymes. Yes? Good question. It's a substrate. You have an enzyme, it's active, it sees its substrate, and phosphorylates it. Now the receptor is activated by the clustering. I have to do this again for you. Here you go. Here's receptors coming together. Picture these as dimers. Okay, now it's active. There's a change. Okay? The point is, it's the clustering that activates the enzyme. It turns out you can experimentally activate these receptors in the absence of the hormone by simply clustering the receptors in the absence of the hormone, proving that the actual clustering activates the enzyme. It's an activatable enzyme. Okay? Question over here somewhere. Yes. Oh, very good question. It's both. It's a both thing. It's not an either or nor. It's a both. It's more or less simultaneous. Yeah, biologically. Now, there's a disease out there called diabetes, and I'll do this over here. There's a type 1 diabetes, which is the insulin dependent, and this is due to beta islet cell destruction. If you don't have beta islet cells, guess